Well, thanks again for the invitation. It's really a pleasure to be able to join you here remotely. And so what I've tried to do is really review with you a set of vignettes that highlight the application of a broad set of materials, but also devices for the extreme environment of the GI tract. And I'm a gastroenterologist, a biomedical engineer. I'm based here in Boston at MIT and also at the Brigham, which is where I practice, as well as at the Broad Institute. So with that, let me dive right in. These are my disclosures. So as part of the work that I'm going to share with you, I am an employee or an affiliate of MIT, the Brigham, and the Broad. I'm a co-inventor of several of the technologies that I'll be sharing with you, and we're very passionate about getting these technologies to help people, and we do that through either collaboration with companies or involved in startups. So these are my disclosures. I'll dive right in. This is my background, and I've been all over, but let me skip this for now. I know in the interest of time. I'd like to just showcase this slide and in particular this photograph on the left. It's a photo I took of my kids right before the pandemic at the Science Museum here in Boston, where there was a bodies exhibit, and this is actually a human GI tract, and I included mainly to remind everybody of the dimensions of this part of our body. My kids at the time were 8, 6, and 3, to give you a frame of reference. But it gives you a sense and really, the dimensions and the challenges, but also opportunities that are enabled by this part of our bodies. And on the right, it's really more of a graphical representation of how I spend my time between these institutions doing what we do. So let me start here on a challenge that was presented to us over a decade ago. And it was really a challenge brought forward to us by the Gates Foundation. There have been many people that have been involved. And here are some of those key folks over the past decade plus. And it's work that was originally funded by the Gates Foundation, also NIH, and then led to the formation of a company that continued to advance the platform. And what we set out to do over a decade ago was really trying to develop systems that were orally deliverable that could provide long-acting release. And that's what you see here, and that led to the formation of a company called Lindra. And at the core of what we were trying to do is really to address a fundamental challenge, and that is of adherence or non-adherence or non-compliance. And what we honed in on was a basic observation, which is that when people are given the choice of taking medication more infrequently, they are more likely to stick with that medication. And I think that perhaps is obvious on the short-term time scale. So within a day, I think most of us, if we have to take medication, would rather take it once a day rather than 3 or 4 times a day. And certainly that's been borne out in several studies. What's interesting is that over the past decade or so, there have been several studies, and I'm showing one out of Japan from 2015, looking at bisphosphonates, which are the drugs used for osteoporosis, where more infrequent dosing was associated with increased persistence or continued filling of those prescriptions. So with that in mind, what we set out to do was really develop an orally deliverable system that can provide metered dosing. And this cartoon taken from The Economist that had been discussing some of our earlier work, I feel captures that general vision sort of very well. And so we did that, and we did that through the development of what you see here, which is a capsule that can basically, you take it, and then as it reaches the stomach, the capsule dissolves and reveals this star-like structure that, because of its dimensions and because of its mechanical properties, can remain in the stomach. And you can see one there in the video. I also have one right here. And basically what we showed, and this is an early experiment in pigs, showing that these structures could remain in place for many weeks if that was the desired outcome. It is also that these systems allowed food out of the stomach, and this is a pig, and I think it's important to appreciate that they're not blocking any food from transit, but also that the tissue in the background that you see is pink, it's healthy, and so it's not causing any abrasions or injury. And so using this platform, what we did was we took that capsule and we loaded different drugs in the different arms of the system. And so we actually looked at three different use cases. This was in the academic realm. We looked at delivery of a drug, ivermectin, that is known to be toxic to mosquitoes, and showed that we could, after a single administration event, maintain desired levels for at least 10 to 14 days. And we then developed a once-a-week antiretroviral system, and then finally developed a once-a-month oral contraceptive system. Now, this work advanced, and so actually today we have a publication in Lancet Psychiatry reporting the phase 3 human clinical trial where these capsules were administered to patients suffering from schizophrenia. And we show the repeated dosing here of risperidone on a weekly basis. And what I hope you can appreciate is we have the immediate release version on the far left in the orange, and then sequentially the weekly dosing. And you can see that the drug levels remain within the bounds of that immediate release consistently in, across this population. And so that really sort of continues, and, we're looking to explore several other applications. Now, out of our early work, we also were introduced with another challenge, and it's the challenge in the pediatric population. And we started to think about ways in which we could administer medications to kids and wanted to explore potential liquid formulations or gel-like formulations to facilitate dosing. And this is work that was led by a very talented postdoctoral scientist, John-Wei Lee, and has been further advanced now in a company, and that's been led by Rahul Danda. And the way we approached this challenge was we really took a step back and thought about, are there unique cues in the small intestine that we might be able to use and build essentially a system around? And so the enzyme that we honed in on is what you see here in the purple, or represented by the purple Pac-Man. And that enzyme is the enzyme catalase. And catalase is known to break down hydrogen peroxide. And so we wondered if we might be able to administer a very tiny amount of a peroxide, much lower than is found in toothpaste or even certain foods, but enough to generate or support the generation of local oxygen to help drive the reaction of dopamine to polydopamine. So let me walk you through here. You have a solution of the dopamine, and basically at the surface of the villi, you have the catalase activity, and that catalase activity is driving that oxygen release. So we have here, again, catalase breaking down the hydrogen peroxide, the liberation of the oxygen, that then is driving that oligomerization of dopamine to polydopamine. And polydopamine is known as a fairly strong mucoadhesive, and it's actually one of the components that is found in the adhesive structures of mussels, the shellfish. So using this adhesive system, we showed that indeed we could achieve this in the small intestine of pigs. And that's what you see on the left here is an everted section of the mucosal surface of the intestine on the far left. And you can see that dark brown layer or paint is actually that deposited polydopamine, which is dark brown. And in contrast, the serosal side, or the outside of the intestine, does not have the catalase activity, and so you don't see that. In the mid portion of the slide, what you can appreciate is a functional deposition of that brown deposition of that polydopamine, given that catalase is preferentially expressed in the upper or proximal small intestine. And on the far right, you can see the quantitation of that polydopamine deposition. And so essentially what we have is a targeted layer that can form on the small intestine. And so we wondered, perhaps we could use this as a platform to add different elements and have different functionalities in this portion. And so we looked at and thought and considered applying enzymes, for example, but also potentially modulating the thickness or the porosity of this layer to potentially transiently block absorption. And that could be helpful potentially in metabolic disease like diabetes and obesity. But also in other use cases, if we had a very thin coating, we could use that to transiently immobilize drugs right at the mucosal surface to enhance the uptake of the medication. And so we actually, we did that, and we immobilized initially beta-galactosidase or lactase, and we showed we could break down lactose much more efficiently And that was very reassuring, at least in the potential, the hypothesis that we had set out to understand and evaluate. With respect to blocking, we showed that we could indeed modulate that porosity. And then if the animal was challenged with an oral glucose tolerance test, that the excursion or the elevation in glucose would be blunted in those animals. I think something that's important to appreciate is that if you repeat this study the next day, then you're back to baseline because the GI tract turns over its surface mucosal surface, the mucus, fairly regularly. And so therefore, one would have to take the medication again the next day. With respect to drug delivery, we looked at the drug praziquantel, which is a drug that the Gates Foundation is interested in. And what we showed was that the exposure or the amount of drug that was absorbed, the AUC, was significantly increased over fourfold. And then by effectively, transiently immobilizing the drug at the surface, we increase the half-life by an order of magnitude by using the system. As I mentioned earlier, this has now been translated and continues to be translated towards human application. And this is work now in collaboration with Synthes, and they've developed solid formulations and pills, for example, and here they show basically that barrier effect being revalidated in pigs, and this continues. And so now they just started a human trial, and the work certainly continues. And I look forward to updating you on that journey in the future. Now, shifting gears a little bit, but also work that we did with the Gates Foundation several years ago, and also continues, is work that's been led by Thomas von Erlach, who is a very talented scientist who developed this in the lab about 10 years ago now, and then has transitioned the work and continues to develop the work at Vivtex, a company we started a few years ago now. And the fundamental challenge that Thomas set out to address is really recognizing that with respect to modeling the GI tract, industry as well as academia has generally relied on models that use cell culture, particularly a cell line called CACO2. CACO2 is actually a colorectal cancer cell line that happens to polarize and look like the small intestine, but it isn't the small intestine. It is a cell line that happens to look like it. But certainly it's been a really helpful tool. But one of the challenges with this tool is that the predictive capacity for absorption lies around 50%. And so what we wondered was, could we do better? Could we develop a more integrated and perhaps representative portion of the GI tract of a large mammal? And our group does a lot of work with pig tissue. And so we wondered maybe we could obtain pig tissue actually from local abattoirs, from so material that's being discarded, but then repurpose that material to develop systems that could support high throughput evaluation of transport across the tissue. And so we did exactly that. We developed the methods for the sourcing, as well as dissection, as well as immobilization in high throughput plates, and then interfaced with high throughput robotics to help do that. We benchmarked our system with about 61 model drugs from the FDA's approved list, really examining drugs across all BCS classes. And what we showed was that our predictive capacity for human absorption was much greater than at least what CACO had demonstrated before. In the very first set of experiments, we selected oxytocin as a model peptide. And what you see on the left in the heat map is essentially about 3,000 different formulations that are derived from ingredients or excipients that are derived from the FDA's generally regarded as safe list, where we agnostically mix and match with the API, in this case oxytocin, and look for transport across the tissue. And the bright green means that there was more that was transported. So we took some of those hits and then we gave them to pigs. And in the first round of selection, what we found was about a 6-fold enhancement orally compared to a control. Now we can do this iteratively and then push that screening. So this is certainly something that Vivtex has continued to do, and now has run tens of thousands, if not hundreds of thousands of formulations through the system to really start to predict and accelerate the development. And here are just some examples. So for example, if you take vancomycin, which typically is poorly absorbed, if you mix it with C10 or sodium caprate, which is a well-recognized enhancer, you can go from 0.2% oral bioavailability to about 1 to 2%. But then if you use the GI-ORA system, what you're able to do is accelerate and identify further formulations that then drive that oral bioavailability to around 50%. So now converting basically a drug that was previously intravenously administered to orally administered. Now this can be applied to a broad range of molecules, and you see two different cyclic peptides in the middle where there have been significant increases. But on the far right, semaglutide, that's obviously well known today as in its as it goes by its other names as Wegovy or Ozempic. And there, for example, we're able to improve significantly over the current orally available, I should say, marketed product by increasing bioavailability about tenfold. So hopefully it gives you a sense on how we're applying ex vivo models to really transform what we can do for the delivery of macromolecules. Well, let me shift gears here to a project actually that was originally funded by NASA about 7 years ago now, and then subsequently by the Department of Defense here in the US, DARPA. And NASA had an interesting request and interest in thinking about how to send microbes to space for a broad range of applications. And then DARPA was interested in looking at traveler's diarrhea. This is work that was led by Miguel Jimenez, very talented postdoctoral scientist. He's now a professor over at Boston University. And we started by really examining the entire field with respect to how microbial therapeutics are being dosed. We recognize that microbial therapeutics come in different forms, whether they're synthetic biotics in that they've been engineered, or they're isolates or consortia mixtures, and then how they're formulated can vary tremendously from capsules to enemas. But also what we recognize is the dosing parameters can also vary a lot, which we believe is likely secondary to the variation in viability in these products. And so related to that, one of the things that Miguel did early on was he went to the local pharmacy here in Boston and bought every single probiotic that was available at the time. And he asked a very simple question. He said, what is the difference between what is on the label and what is inside of the bottle? And what you see here is essentially a representation of that, of the total number of cells and of the promised cells, right? And you can see for some of the products, they're close, but for many, they're quite, there can be a big gap. And this was really important, really, in informing and telling us, there's variation, and likely shelf life will also play into the viability of the products, but also the potential to have a clinical impact or effect. And so what we set out to do was identify materials or ingredients that could help stabilize microbes through a stressful process. And the stressful process here was lyophilization. And what we found was we took 3 bacteria and 1 yeast and put them through lyophilization mixed with a library of ingredients from the FDA's generally regarded as safe list, and found multiple ingredients that could stabilize these microbial therapeutics through the stressful process. We then kept these systems, or I should say these formulations, at room temperature out to a month. And we found a few that could keep essentially the organism stable even out to a month at room temperature. We also started to characterize the landscape of these microbial therapeutics. And what we can see is that for some of the organisms, there's a favorite or a preferred set of ingredients, but we also find certain ingredients that are, quote unquote, universal stabilizers. Although in our experience, we tend to find the preferred set to be superior to those universal stabilizers. And so we matured some of these formulations, very specifically ones that were stabilizing E. coli Nissl. And I know that this is a product in Europe and used for many different applications, from diarrhea to irritable bowel syndrome and inflammatory bowel disease. And so this was now in the context of working towards therapies for travelers' diarrhea. And we took E. coli Nissl and we showed that we could stabilize it, and initially at room temperature out to a month, whereas the commercial product, Mutaflor, will die within a few days. But then at 37 degrees, we showed that we can maintain significant viability, well beyond 6 months of 37-degree incubation. And again, Mutaflor, the commercial product, will essentially disappear or not be viable within a few days of incubation at 37 degrees. Now, as I mentioned to you, part of the work was supported by NASA, and they're interested in looking at many aspects of human, I would say, space exploration. It certainly includes health, but also other practices like potential agricultural practices. And so what we tend to focus on is really applications and thinking about how these potential materials interface with formulation or development of different tablets. And one of the key aspects that we looked at was whether these new formulations could stabilize the microbes through fairly stressful processes like milling, which exposes these organisms to significant compression and shear forces, but also wet granulation that includes the exposure to organic solvents, and then also tableting, which also involves the compression coupled now also to organic solvents. And what we found was that indeed our formulations were much more stable than some of the excipients that are used commercially. Additionally, we looked at resistance to ionizing radiation. This is in the context of space travel. And one of the organisms we were looking at is actually an organism that is known or used sometimes in plant science and known to induce nodule formation in the root structure of plants. And those nodules are associated with nitrogen fixation. And we had exposed our formulations to 50 degrees C to see whether the formulations could still withstand sort of that temperature and preserve the viability and function of the bacteria. And we showed here functionally that indeed after 50 degrees C that we could still induce those nodules. And then with respect to traveler's diarrhea, in a loop model in pigs, we showed that indeed we could still reduce Shigella levels using the E. coli Nissl that had been formulated in the more stable formulation, even after 37-degree incubation. So let me shift gears here now to a more device-centric sort of approach on drug delivery of challenging molecules. And this is work that started about a decade ago. There have been many people involved over the years, but these are some of the key contributors over that period. And the core challenge that we set to address is the one that I'm showing here, which is, in general, for medications like insulin or drugs like adalimumab or Humira, those medications generally require an injection. And although the majority of people, if they have to take medication, would prefer to take it orally, the challenge, of course, is that drugs like insulin are very poorly or not absorbed at all through the GI tract, and or are readily degraded by the environment in the GI tract, both the proteolytic sort of activity of enzymes there or the acidic environment. And so one of the big areas that we focused on over the past decade has been developing solutions to address this. And so this was largely inspired by some early experiences that I had as a gastroenterologist going back to 2009, 2010 or so. And these are two real cases, people that I looked after at the Mass General, which is one of the hospitals here in Boston where I trained. And the first case is a gentleman in his early 40s that had been transferred from a local jail. And he had been transferred having ingested shards of glass. And so it was a Friday, and I was the fellow on call at the time, and I was called to come down and see this patient. And as I approached him, I could see that he had a half-eaten sandwich in his hand. And, he actually looked comfortable. I asked him to stop eating. I examined him, and his abdomen, his belly was soft. But because he had eaten, I knew that I wouldn't be able to help, for example, with an endoscopy. I examined his studies. He had an X-ray. I could see three small objects on the X-ray. And I was at the time I was a gastroenterology fellow, and I called my consultant, who's a senior gastroenterologist, and I reviewed the case with him, and he said, "Well, we'll have to observe him, because he had just eaten." And and that's exactly what we did. We observed him over the weekend, and I was on call that weekend, and so I would come in, he had an X-ray done every morning. I would examine him, and his abdomen was soft, and I could see over the course of the weekend that these objects were sort of moving through the intestines, through the bowel. And by Monday, they were gone, and he didn't even notice. This, to me, was a mind-blowing experience in that it really showcased the capacity for the GI tract to tolerate sharp objects. I think what's important here, and what really drove the decision to observe him as opposed to consider something more emergent like surgery eventually, was that the objects were 2 centimeters or less in their maximal dimension. And that number is very important because it is the dimension that we recognize for objects to actually being able to transit safely through the GI tract. I presented this case along with other cases at our grand rounds at the time, and this to me was a very sort of formative experience and case. Now, the second case that I want to share with you is a more common case that we tend to see. And this is a lady in her 60s who has a history of arthritis, who has been using a lot of ibuprofen. And ibuprofen is a wonderful drug, but has some known side effects. And she comes in complaining of epigastric pain. Epigastric pain is pain overlying your stomach, but also reporting that her stools were dark and tarry and that she was lightheaded. And so that sort of constellation of symptoms and signs and the history really makes us think about gastric ulcers. And indeed, in this case, we did an endoscopy and found an actively bleeding gastric ulcer. We injected that with epinephrine, with adrenaline. And we do that because adrenaline has a local vasoconstrictive effect. And so meaning it clamps down all the arteries, and it allows us then to clean the area and have a look. And then we can intervene either with cautery or clips. But as soon as we give that injection, what we also know is that all the alarms in the room start bing-bonging because that drug that we gave for a local effect circulates through the body and then acts on the heart. And so putting these two together, we created these capsules. We had this idea, maybe we can create capsules that can give you an injection from inside. And published a small paper describing this concept. I was able to get some free samples of insulin. My wife is an endocrinologist, and she was kind enough to help me with that. And I injected the GI tract of a pig with insulin and showed that we could induce reductions in glucose levels. And also showed that capsules with many needles, such as the ones that you see on the slide, could transit through the GI tract of a pig without causing any damage. So this was really, I would say, presented the idea, the hypothesis, but there were still some major challenges in that if we wanted to create an ingestible system that was capable of administering drug, we would need that system to engage the mucosa in a predictable fashion every single time. Two, we would need it to trigger or inject autonomously, and somehow we would need to administer enough drug for it to be meaningful, while still being small enough for it to be able to transit through the body safely. And so we took a page out of the self-orienting leopard tortoise, or weevil wobbles also, and generated these capsules that have the ability to self-orient, as you could see there in the animation. Now, we tested these systems with high-speed photography and different media, and then also in the stomach of pigs. And what we showed was that we could achieve self-orientation reliably and in spite of active or vigorous movement in the stomach. So this was very reassuring on the self-orientation and ensuring that we had good engagement. The next challenge was, well, how do we trigger an injection event autonomously? And so somehow we needed to identify a way of maintaining, let's say, a compressed spring with a needle and then having that spring trigger autonomously. And so what we did was we identified a carbohydrate. In the very early days, it was a sugar glass, and then later isomalt that can constrain or maintain a compressed spring compressed, and it is sensitive to the human environment of the stomach. And we can very tightly control the timing of the release as a function of the circumference of that cylinder. And what you see here is high-speed photography of one of those sugar encased springs in a humid environment. And the reason I have it here repeating, and I just want to highlight, is that this is taken at 10,000 frames per second. And what you see is that sudden release of energy sufficient to give that injection. And the reason that this is important to appreciate here is we are using a material that has brittle fracture mechanics, which allows or enables that very rapid sudden release of energy. And then with respect to administering enough drug, we created solid needles of insulin. So typically most biologics are administered in the form of a liquid. Now, because they're administered in a liquid format, they're typically constrained by the solubility limit of the solvent, essentially on the API and the drug. And so what we wanted was perhaps we could create solid needles, maximize our loading efficiency, and thereby maintain or preserve sort of the size of the device. So we were able to get large amounts of insulin and compress it into small needles. And then what we showed was that we could administer those through the GI tract and achieve the comparable levels as when administered through the skin. We also conducted several safety studies looking at the depth of penetration, initially in pig stomach, but then later in even cadaveric human stomachs, and showed that we could control and maximize the safety by minimizing essentially the insertion and penetration depth. We built on this work and demonstrated that we could also develop capsules that could deliver liquid. And certainly for certain applications, having a liquid formulation can be really important. And so there, what we had to do was define and identify the optimal depth of insertion of the needle in order to achieve a submucosal depot. And that's what you see here. In the bottom, you see a micro CT image of that depot being created. And next to it, you have a cross section with showing you the tissue and that depot in place. And the way that we did this was by using a two-spring system where we have an initial injection event that injects the liquid, and the second spring essentially retracts that needle after the injection takes place. Using this system, we demonstrated that we could deliver drugs like Humira, insulin, GLP-1 receptor analogs, as well as adrenaline. Now, using this approach, and I should say we did this work before the pandemic, but we also demonstrated that we could deliver mRNA and showed that we could do that in pigs. In order to do this, we had to develop a polymer-based nanoparticle system to help carry the mRNA. And we demonstrated that using a polybeta amino ester-based system to carry the payloads. And we demonstrated that in pigs. Additionally, with respect to the platform, this actually has gone now into humans for an initial safety study, and I look forward to sort of updating the community on that soon. Additionally, one of the things that we always wondered when we started developing these systems was, maybe, is there a way that we might be able to eliminate the need for a needle at all? And could we use a jet from these systems? And so inspired by squid and cephalopods, we actually set out to define jetting in general in the GI tract, and we created a family of devices, one similar to the other ones that I've shown that self-orient, but also others that can essentially send the projectile from the side to really help support administration in tubular organs. And so both radially and axially emitting jets, and that's what you see here. Now, just to give you a flavor of what this looks like, these are the self-orienting systems. And what we have is here 4 different test systems showing you sort of those jets in profile, so you can see that occurring, and you can see the delivery here in a model membrane. And using this system, what we had to do was characterize essentially the pressure that was optimal to achieve the depots in the different parts of the GI tract. So we characterize every part of the GI tract in the large mammal. And here I'm just showing you representative data sets where I show you in cross section the micro CT of that sort of depot with the parameters that we had identified as being optimal for that part of the GI tract. And then using the system, we showed that we could deliver drugs again, like GLP-1 receptor analogs, as well as RNA, so siRNA using these systems. And we showed that the levels that we could achieve were comparable to those that are achieved with an injection. Now, I want to end on something a little bit different and just, I think, for some food for thought, a little bit outside of the material science side, but certainly I think material science has a really big role here as we continue in this area. And it's the idea or concept of ingestible electroceuticals. So I've talked a lot about drug delivery science, and what I'd like to introduce here is the potential to have ingestibles that are not necessarily releasing a drug, but are releasing a current where they are essentially zapping or stimulating the GI tract as the mode of therapy. And this is work that was inspired by observations from people who have these. This looks like a pacemaker, but it's sitting in the stomach or in the abdomen and connected to the walls of the stomach. And it's a pacemaker-like system that's used for the treatment of this condition, gastroparesis. Gastroparesis is a condition that is sometimes associated with diabetes, where the stomach isn't moving quite right. It's moving more slowly. And so given that, we wondered, whether, these systems, well, actually, let me take a step back. One of the challenges with these systems is that the effects with respect to prokinetic effects, meaning how they stimulate, are not that pronounced. And in fact, I can share with you that we, for example, at my hospital here or the previous hospital I worked at in Boston, did not place them in patients. And it's because some of the functionalities perhaps are not as good as one would wish they were. But what's interesting is that when one looks at retrospective data in people who have had these implanted, the patients report feeling much better. And this is really interesting in that they report feeling better, reduction in nausea symptoms. So we wondered, perhaps that electrical stimulation is actually inducing release of a hormone or hormones that are changing essentially how the person is feeling. And so we actually repurposed the tool that you see here under A. It's called the gold probe. That is actually the tool that we also use to cauterize or treat ulcers when they're bleeding. But we repurposed it, and instead of applying the electrocautery current, we changed it so that we would just stimulate without causing any tissue damage, excuse me, or any coagulation. And we actually essentially applied the parameters that were derived from that pacemaker system, the Entera system, and showed that we could induce releases in the hormone ghrelin. We then optimized the parameters as far as the amount of time that stimulation would occur for. And at least our vision was perhaps we might be able to create a capsule that can apply this current in the stomach to induce ghrelin. Now, the challenge with trying to do that is that there's always some fluid in the stomach, right? And so I include this photo just to remind me, really, and to remind you that there are essentially puddles in the stomach, right? And so how do we apply a current and avoid or minimize parasitic losses of that current into those fluid puddles? So we took a page out of this animal that is called the Australian thorny lizard. And it's interesting because one of the ways that it captures water is by wicking it using microfluidics in its on the shell or outside on its skin. So we said, maybe we can do something similar. And so we created capsules with tiny grooves so that they could wick the fluid, but still keep contact at the tips of of those grooves. And so using these systems and placing essentially the electronics that could actually apply the stimulating parameters that I had described earlier, we created these capsules right here. And then we gave them to pigs, and we showed that indeed we could induce the hunger hormone ghrelin using these systems. So with that, I want to thank you for your time. I'm happy to take questions. I want to thank our team. We have our team members here along with our family members at our summer gathering last year. And with that, I also want to share with you my email. If you have questions, you want to reach out with anything, feel free to email.